The research question we have in my group is best illustrated using colors. So as you all, I think, appreciate, flowering plants come with enormous diversity. So they form different, they have different colors, different scents, different shapes, which nevertheless raises the immediate question, how does this enormous variety evolve? And of course, Uppsala is a great place to address this question because in Uppsala the foundation of modern taxonomy has been laid by Carl von Linné. And of course, we're also not the first ones pondering about how plants evolved. It goes way back to Charles Darwin, who in fact was rather annoyed by the fact that there are so many flowering plant species, because what he realized is that flowering plants arose to ecological dominance in a relatively short time period, which was difficult to reconcile with this idea that speciation is a process that takes long periods of time. So this didn't really look like a long-lasting process, but rather like an abrupt event. And because he was lacking a satisfying explanation for that, for him it was the abominable mystery. And what we found is that there are indeed mechanisms that very rapidly drive plant speciation. And these mechanisms take place in the endosperm. The endosperm is a tissue that sustains embryo growth. And it does not only sustain embryo growth, it also sustains us, because the majority of our daily calories are derived from the endosperm. So I think it's worth to take some time and invest some efforts to understand how the endosperm actually works. And the endosperm is a very peculiar tissue. It derives after fertilization of a diploid central cell. So after fertilization, it's a triploid tissue, having 2 maternal and 1 paternal genome copies, different to most other tissues. And this genetic peculiarity causes also special mechanisms to take place, epigenetic mechanisms that either act directly on the DNA in terms of DNA methylation or on the histone level. Histones get modified. And this causes certain genes to adopt a very peculiar expression pattern. It causes them to be either active only when they are maternally inherited, so only the maternal alleles are on, or only when they are paternally inherited, so only the paternal alleles are on. And these imprinted genes are very important. What they drive actually is, or they have a very important impact on endosperm development. And just to give you one example, so when you cross 2 different species, or when you cross the same species, so species A with species A, you obtain viable seeds, you have a nicely developed embryo. While if you double the chromosome number in the species, you can very easily do that, and in nature that happens on a very frequent basis, and you now do a cross, so species A with the species A with doubled chromosome number, all seeds will abort. And they abort because the endosperm does not develop properly. And when we now knock out imprinted genes in this polyploid species, what will happen is that all our seeds are viable, and all embryos can develop normally. So with other words, these imprinted genes establish hybridization barriers in the endosperm. And these genes are very rapidly evolving. So that means they can very rapidly establish hybridization barriers, explaining rapid evolution of new plant species. And we would like to drive that further. In the future, we would like to identify the functional role of these imprinted genes. We know that they drive endosperm proliferation with that seed size, so of course this is of applicative potential. We would like to understand the mechanisms understanding these barriers in the endosperm, and we would also like to develop epigenetic tools bypassing hybridization barriers, easing crosses between different species, which for plant breeding, of course, is of value and importance. And we have already nice tools, so when we remove DNA methylation, we can completely bypass hybridization barriers. And of course, I would only like to show that this is a team effort like science is today. So thanks for your attention.